Get started. Let's see if that worked. Yes, recording in progress. Great. Okay, welcome everyone. Good morning. This is Agential Topics. I'm Charlie Munford. Agential Topics is a periodic study session put together in part by Talking Octopus, which is a nonprofit organization that is dedicated to building a physical model of biological agency. And to that end, we have periodic sessions where we try and discover what biological agency really is. So that's the meeting you're in, Mr. Gart, and this is an informal group, wide-ranging discussion, but very focused on biological agency.
Wow.
So yeah, so today I'm gonna slightly break with the tradition of not introducing our guests and uh, and say a word or two about Denis Noble, who's our guest for today. Just because, in addition to being a very generous and broad-minded scientist, he's some would say the founder of systems biology, and so we're very lucky to have Denis with us today, and I think he has some slides. And the format will be that Denis will present some slides, and then I'll ask a few introductory questions to sort of open up the discussion, and then we go from there. And you can raise your hands or just jump in. Try to stay muted when others are talking or when the slides are going. And if you don't stay muted, I may intervene and mute you proactively, but realize that if you have something to say, be sure and check your mute button. So thanks a lot, guys, and off we go.
Okay, well, thank you very much. And thank you, Charlie. As you know, I'm quite ancient nowadays. I even, other people in this group won't know this, but I even taught Charlie Munford's father. There you are.
Uncle.
How old can you get? Anyway, I will now move to um, sharing my screen and go straight in. So what do I need to do here? Yes, I will share my desktop. There we go, and go straight to the presentation. And appropriately for this group, I've had the audacity, if that's what you want to call it, to use the title, How Does Agency Arise? And I'm going to argue that it does through life itself creating robustness and meaning, and also by showing that gene-centered biology particularly has confused two very important philosophical concepts, association and causation. They are not the same, and that's a key feature of what has just appeared, which is this book published this month, but being distributed towards the end of the month, The Pacemaker Channels of the Heart, which is in fact where my entry into systems biology came from, because I was the person who created the first model, biophysically based model of the pacemaker channel work of the heart and its result in creating cardiac rhythm way back in 1960, so 2/3 of a century ago. And within that book, there's a lot that deals with the question of robustness. I also highlight two very recent articles that will be central to the presentation. First, the article in Evolutionary Biology by myself and my brother Raymond, How the Central Dogma and the Theory of Selfish Genes Misled Evolutionary and Medical Sciences in Understanding Multifactorial Diseases. Then the second is a shorter review, the Cardiac Pacemakers, a paradigm of robustness in evolutionary biology, which has the essence of the argument in the book, The Pacemaker Channels of the Heart. And I'm going to divide the presentation into two relatively short parts. Part one will be how robustness evolved and why association is not causation. I'm also going to do that because without first of all demonstrating the flexibility that is necessary in order for robustness to evolve, you won't be able to understand the second half of the talk, which is how does agency arise? So let's go straight on. How robustness evolved in the heart is a key to all of this. The rhythm shown there at the right in that slowly varying rhythm of the heart model anyway, but based on detailed experimental information, that is generated by multiple functional networks in the diagram on the left, which is actually the abstract diagram for the article in the Journal of Physiology, because the rhythm is generated by those multiple functional networks of interacting ion channels, which are proteins in the membranes of the heart, cell metabolic processes, and the membrane electrical processes. And what the bottom part of the diagram on the left shows is that there are at least 4 genetic components that can enable the network to generate rhythm, provided any one of those is present. Normally, all 4 are present. They therefore can be seen as backups for each other. That's the logic of that diagram, that any one of that polygenic input is sufficient to ensure that rhythm will continue. Now, this is from the paper that demonstrates the basis of robustness in the case of the heart, and then I'll come to generalizing the idea. What we did in this paper, it was published over 30 years ago now in 1992. The top diagram is the rhythm diagram you saw in the previous slide, very gently slowing of the cardiac rhythm. How did we generate that? We played with 2 of those 4 genetic components or their proteins. The middle one is a mixed sodium and potassium channel protein, and the bottom one is a sodium channel protein, not yet actually identified genetically, but it certainly exists. We can record it electrophysiologically. Both of them contribute to cardiac rhythm, together with, remember, many other components in the functional networks. And the experiment we performed here was to progressively block the component generating most of the current generating the rhythm. That was the sodium channel at the bottom, down to 80%, 60%, 40%, 20%, and then complete knockout. And what you'll notice, which is very surprising indeed, if that is the real cause of the rhythm, is the rhythm continues with just a very gentle slowing. And the reason is also obvious. What you can see in the middle trace there is that as you do that automatically, with no input from the whoever is manipulating the program, with no input other than this change in the progressive block of the channel conducting just sodium, the other channel just kicks in. It does so largely because of the changes in voltage themselves. So there is a complete interlocking between the networks using the 2 genetically based or protein based components. You can do a very simple calculation now. The actual change in frequency there is somewhere between 10, excuse me.
Looks like something's gone wrong with your audio, possibly, Dennis. Is that possible?
Sorry?
Oh, there we go.
That's better.
There we go.
Now, the key is that the association score you would get between the component, the sodium channel, and the change in frequency on a complete knockout is only between 10 and 15%. But the causation in terms of the electric current, sorry about this, is actually around 85%. So you've got 85% causation demonstrated physiologically as the current generating the rhythm, but only a 10 to 15% association score. Now the question is that problem general? Well, it is. This paper, published nearly 20 years ago by Hillenmeyer and his colleagues in Science, showed that in yeast, over 80% of gene knockouts are actually silent. There's a zero association score in normal physiological conditions. But they also went on to show, again, parallel with what we showed in the case of the heart, that most of these show functionality in times of metabolic stress. So you've got the same kind of robustness. If this genetic component or protein is not used and cannot be used, is absent, then others take over. And this all explains two things about genomics. First of all, most of the genome association scores are very small. So much so that what people have done in order to try to arrive at genetic causation is to create what are called the polygenic scores, which means that if you've got, for example, 300 genes associated with, say, a disease like schizophrenia, you add all the association scores together. Now, a very simple point that many of the mathematicians in this group will understand immediately is that in a complex system network, that is not valid anyway. But even if you do it, does it work? This article by Hingarani and his colleagues at University College London was designed precisely to ask that question, because what they did is to take the polygenic risk scores stored in the National Health Service database here in the United Kingdom. We've had now over 20 years of genome sequencing, but also at the same time, the recording by the National Health Service of the diseases that those people eventually suffered from. Cardiovascular disease, cancer, schizophrenia, Alzheimer, you name it, they're there. All the information is there. You can therefore ask the question, do the polygenic risk scores predict the risk and the actuality of occurrence of those diseases? And there's just a quote there from their paper: polygenic risk scores perform poorly in population screening, individual risk prediction, and population risk stratification. They then conclude strong claims about the effects of polygenic risk scores on healthcare seem to be disproportionate to their performance. This is typical for the American listeners here, typical English, what should we call it? Um, uh, playing oneself down a little bit. It's actually appalling. It's actually shocking that after 25 years we don't have a better outcome than that. So now I come to what needs to be done. Well, frankly, what we need to do is at last something like 60 or so years late, but better late than never, to replace the role of the central dogma of molecular biology. On the left there, I've summarized what Crick's formulations of this were. First, his original direct 1958 formulation, DNA creates RNA, which creates protein. And then the 1970 formulation, which admitted that there were two exceptions to the straightforward arrow DNA to RNA to protein. First of all, back transcription of RNA to DNA, and self transcription, if you want to call it that, self replication of DNA and RNA. Now, you'll notice that no other parameters in the physiological system in which this is all embedded was ever considered to be relevant. Well, the diagram on the right is what Raymond, and I published first in the journal Evolutionary Biology just earlier this year. And what you'll see, of course, is that there are numerous ways in which those processes can be influenced. Nothing is going to lead to protein being used directly to make RNA or DNA. That was the mistake, I think, that was made by Francis Crick and others in the interpretation of the dogma. It was as though it had shown that in no way could organisms in some way change their genomes to produce new genomes if they needed to. Well, that's just rubbish. But there's something else that's also rubbish here. You'll notice that the self-replication arrows, DNA to DNA, RNA to RNA, those very strongly curved arrows going back on themselves, those were presented as solid, complete, accurate self-replication. We now know that is not true. That's why in the new diagram on the right, I have lightly dotted those arrows to indicate that those processes are only partial self-replicators. We'll come on to how partial in just a moment. But then I've also put in, together with Raymond, all the various other processes, including the functional networks, where the robustness that I described in the heart and in yeast, and then through genomic low association scores, to occur generally in relation to biological functions. So I think we have to conclude. Regretfully, if you had great hopes for the genome sequencing project, DNA is simply not the blueprint for life. Incidentally, that's an article that I published in Nature last year, by invitation of Nature, incidentally. So, what physical and biological features are essential for purposeful, meaning-making organisms to exist? Well, my argument is simple, and it's summarized here. If DNA was the blueprint, then responsible agents could not possibly exist. We would have a complete mechanism. Now, DNA does not even begin to have the necessary properties. First, and this has been discovered only in the last 20 years, it is in fact a very poor self-replicator. That's the reason those arrows in the diagram on the previous slide are dotted. In a genome of 3 billion base pairs, self-replication, meaning nucleotides finding their corresponding base in the unfolded double helix to create two new double helices, that kind of process would achieve an error rate of around 1 in 10,000 base pairs. Now that may seem pretty good. Chemically, it is. But physiologically, it's far from adequate. There would be hundreds of thousands of errors if that was the sole process of replication. So it cannot be. So a fundamental aspect of Crick's diagram is actually incorrect from the very beginning. Those solid arrows are misleading. Moreover, chemically, DNA is actually a very inactive molecule. It can be read, it is read, but it cannot do anything else. The really active molecules in cells are the RNAs produced from DNA, which can also be enzymes, and the proteins. And finally, what we've learned from the previous studies that I've summarized, association does not prove causation. Incidentally, it's also the other way. Those zero scores in the Hillenmeyer study in yeast do not mean that those genes did not have a function or had any causation. So even a zero association hides or can hide substantial causation. So I now come to the second part of my talk, how organisms therefore harness the stochasticity in their water-based environments, the environment, of course, of all cells of the body. Because water, as many of us know, is a very unusual chemical. The range over which it is liquid is important. It's the range of terrestrial temperatures on Earth. And my demonstration that we cannot be determinate machines starts, therefore, with two of the key four elements of which life is largely based. Hydrogen and oxygen are those two. Combined, they make the smallest and the great majority of all the molecules in our body. And this is, of course, liquid at a temperature at which the individual atoms would be gases. I come to now another very simple calculation to throw into my talk. Take the smallest living organism, a bacterium. It's around 1 by about 4 microns. There are 20 billion water molecules in that small space. I challenge anybody to produce the possibility of any conceivable available computer that we can build that would be able to reproduce the innumerable water states in such an organism. Because remember what Robert Brown showed in 1827. Water molecules are continually jiggling around in a stochastic fashion. We call it Brownian motion for that reason. And it was Einstein in 1905 who showed that that process in moving particles around suspended in water is in fact the result of the bombardment between the water molecules. Incidentally, this bacterium can swim for where it wants to go with all of those cilia. It knows how to find its food. It's intelligence of a kind in a very tiny volume. So what do organisms do with this vast stochasticity? Well, they use it. You see, the fact that DNA cannot self-replicate, it can't replicate like a crystal, to quote people like Richard Dawkins, even though it's true in part, is based on a very simple chemical fact that if that were all that we could use, there would be around one error in 10,000 base pairs. A human genome of 3 billion base pairs, there would be around 600,000 errors. And this makes, and this is the interesting point, this makes massive stochasticity available to every living cell. And the question then is, how do they use it? Well, first of all, living cells correct nearly all of those errors. The error rate allowed by a living cell before it allows itself to divide, it actually waits for the error correction process to occur. That's orchestrated by the living cell, incidentally, orchestrating a number of cut and paste enzymes to creep along the DNA threads and get the error rate down to an error rate of, wait for it, 1 in around 10 billion. And the cell only divides when it knows that that error rate has been achieved. Because if it divided before then, in a multicellular organism like us, it would be destroyed by the immune system long before it got the opportunity to reproduce itself. And that means, this is the key, that since that is all under cellular control, cells have got the ability to control how accurate that replication is. And if you ask the question, what was the immune system doing during the pandemic? It used that stochasticity and the fact that if you downregulate the error correction, you can get a vast number of new DNA to precisely do that within a very small part of the genome coding for the variable part for an immunoglobulin. The immune system achieved hundreds of thousands, if not millions, new possible DNA codings for the variable part, the grabbing part of the immunoglobulin. I argue also that other systems in organisms like the nervous system can also use and harness this kind of flexibility. And incidentally, it's Alan Turing himself, the godfather of artificial intelligence, in his 1950 paper who wrote it is probably wise to include a random element in the learning machine. He knew even way back then that AI would not succeed if it did not harness stochasticity. And he says, since there is probably a very large number of satisfactory solutions, the random method seems to be better than the systematic. And he even went on to notice, this is published in the philosophical journal Mind, it should be noticed that it is used in the analogous process of evolution. Even way back then, when Crick was about to formulate his automatic central dogma, there was Alan Turing noting that evolution doesn't work that way. I love it. Now, my conclusions. Living organisms precisely control the openness in their stochasticity. That's the basis of their robustness. It's the basis also of their ability to change when they need to. And each organizational level, this is the principle of biological relativity for those who are familiar with that idea. Each organizational level achieves that by harnessing the stochasticity of the level below. This is precise multi-level meshing, every level meshing with the level below it and controlling it. Because once harnessed, and this is the key to what organisms are doing, randomness is no longer random, it's chosen. Meaning maker, therefore, meaning making, therefore arises from the harnessing, leaving the system open to times improbable, but certainly rarely so necessary behavior when that is required. And so I conclude, I want to thank my co-author of Understanding Living Systems, a very simple book explaining all of this for non-biologists, published in 2023. And I also acknowledge very valuable discussion and comments by my colleague in France, Ren Bourret, on the relevant publications that I listed at the beginning of the talk. So thank you very much for this opportunity to talk to this particular group on agential, forgotten what the title was, agential systems, yes. I think living systems are clearly that, and I think we are beginning now to get a glimpse of how it is that they do it. And with that, I'll stop and stop sharing my screen if I can find where I have the way of making the change. Where do I do that? Here?
It should be at the bottom. There's a green button.
Ah, well, that's the problem. You see, I've lost myself, haven't I? Oh, here we are. Stop share. Great. There we go. Over to
Fantastic.
you, Charlie.
Okay, Dennis, thank you so much. That was beautiful, wonderful, and such a concise and particular picture of your general philosophy and your general work. And I know many of us in the group have spent a lot of time with your books. And some of us were talking earlier, and we were saying that, Denis Noble is a person who causes tachycardia in many of us. So, so anyway, yeah, I wanted to open up the discussion to kind of anchor this presentation and your bigger thesis. So, what you're talking about here, harnessing stochasticity, you're more or less demonstrating that the selfish gene theory can't be right because DNA doesn't self-replicate, and the replication process, as well as the gene expression process, is controlled by the higher level physiological constraints.
Indeed, yes.
Yeah. So then you sort of go from those conclusions to the bigger picture of biological relativity, which is that multi-level causation is ubiquitous throughout biology. Um, so my main question about that, I mean, I think most of us in the group are pretty on board with the contradictions in the selfish gene theory, and pretty much the group is founded around, the idea that that's incoherent. So we're fully on board with that, but part of the aims of the group is to sort of uncover the alternatives and the particular predictions or conclusions of alternative views of how order might emerge in biology. So I wondered, my biggest question about your work is those physiological constraints that bind stochasticity or determine precise sequence. By sort of, as you've in other places you've described it as a fruit machine, which us Americans will know as a slot machine, the type of machine that mixes options, and you can pull the lever and arrest an option and fix it in place, and then scramble the next one and fix it in place. And you talk about the way the cell does that. The immune system cells do that when they're making antigens, antibodies, sorry, immunoglobulins. So that process of arresting stochasticity or binding stochasticity that comes from the higher level to the lower level implies a sort of, orderliness or a lack of entropy. And the paradigm view, the neo-Darwinist view, is that all the entropy in living systems is purged through natural selection, that there's no other sort of selective process or creative process. And so we're all familiar with the logic of that argument and why it can't be true. But the question I have for you is in your argument, there seems to be a source of order that's coming from the higher level physiological constraints and being impressed upon the stochastic level below. And we can see that at other levels, the way that individual organisms can fit into a community of become a superorganism, like an ant colony. Um, and so the question is, where does that order at the higher level come from? Is it the way I imagine when I read your work, I imagine what you're saying is that the order in physiological systems comes through the natural selection of those physiological systems. And I wonder if that's the complete extent of your answer, or there's more, there's a whole.
No, it can't. I think you're bang on, Charlie. It can't be. Um, you see, the some people were familiar with the relevant argument here in this group. The infinite monkeys theorem won't work. That was raised in the 19th century in the course of the discussions of Darwin's and Wallace's ideas on natural selection. And incidentally, Charles Darwin himself was completely convinced that natural selection would not do it alone. And his argument was also the same as mine. It's far too slow. Something has to be speeding evolution up, and that has to be, relatively speaking, intelligent. Now, he chose two or three other mechanisms which he thought existed. The first one was that just as he had compared natural selection metaphorically with what he called artificial selection, which is the deliberate choice by a breeder like a human to breed certain varieties of animals rather than others, just as that can happen, so other organisms can be exercising the same kind of choice. And he gave it to the birds and, of course, to other organisms that he thought were capable of it. He called it sexual selection. My preference is to call it social selection because it doesn't actually restrict itself to whether the cooperation is to help the plan of organisms, its group, to survive, or whether it helps it in reproduction. Either way, it is helping the evolution and survival of the group. So the first thing to notice is that Darwin did not think that natural selection on its own would be enough. And we now know that that is correct. It's been shown by two mathematicians that the infinite monkeys theorem doesn't work. They, I perhaps will, I could I leave this to whether people want to take me up on that? Because if I try to explain what the infinite monkeys theorem is and then why it doesn't work, I get into some maths which, first of all, I don't have a screen to write on, and second, I don't think we need to go into that kind of depth unless we really need to. The fact is that on its own, it would take um, it would take billions of years just to get anything from the infinite monkey idea. That is, a monkey typing randomly on a computer keyboard to generate anything that's meaningful. So let's just take that for granted for the moment. Darwin fully appreciated that, and not only did he think that it was the case that other organisms than humans could also exercise choice, an idea which incidentally the neo-Darwinists have excluded completely. It's not in the modern synthesis, deliberately excluded. The other thing that he favoured was precisely what Lamarck had come up with in his ideas on evolution 50 years before Darwin. And incidentally, Lamarck drew a tree of life, the transformations of species into many different varieties, something like 28 years before Darwin ever drew his Galapagos island tree. So, not too surprising, Darwin knew about Lamarck's work. He actually praises him in the fourth edition of The Origin of Species. And he also assumed that inherited characteristics could be passed on. But that gives you billions of years of knowledge generated by organisms. Add to that what I call the ratchet effect. When you have passed a particular threshold point in an evolutionary process, and there are many, you can't go back. So there is a one-way transition. I will just give one example of that. The reason for the existence of organisms like us and for the existence of a heart consuming a lot of energy to produce its mechanical beating is precisely the fusion of one organism with another on the process called symbiogenesis. This was championed by Lynn Margulis, but it was also previously um, shown as an idea for the origin of mitochondria in particular by two Russian scientists in the very early part of last century. So for over 100 years, we've known of the existence of this process. Why do I say that's absolutely essential and was a ratchet point in the process of evolution? The energy that you can get from a mitochondrion processing the chemical transitions to produce ATP is to produce 10 times as much ATP as would have been possible before that fusion. That energy was needed for multicellular organisms like us to even exist. What I'm saying here, therefore, is that by using the combined knowledge, wisdom, if you want to call it that, of the many aeons of evolution and embedding it in the future through both inheritance of acquired characteristics and in the intelligence of organisms to choose each other and who they mate and cooperate with, you have a totally different view of the evolutionary process. In fact, I would go as far as to say that evolution today, and for the last at least 500 million years, since the existence of organisms capable of this kind of choice, we have not had pure natural selection. Most of the other environment that is influencing us and all the other organisms on Earth are other organisms with this ability. Once you accept that, you've got masses of intelligence to play with. So I think my short answer, um, uh, Charlie, to your challenge is I don't see the difficulty. I haven't done the maths necessary to prove that all of this can work, though I do know that the infinite monkeys theorem, which was the idea that you could get it totally randomly. It just doesn't work. You can't get it randomly, and you're quite right on that to highlight that difficulty, Charlie. My answer to that is that's not how it happened. It could not have happened that way.
Right. So there's sexual selection and also Lamarckism is based.
Yes, I think you have to bring those both in. And I think that if you reflect on how both of those were excluded, Wallace did so in 1866. That's when he departed from Charles Darwin, just 7 years after they had published together at the Linnean Society on the idea of natural selection. Wallace was convinced. He didn't know, of course, of even the possibility of doing a calculation to show whether an infinite monkeys theorem would work. But he imagined that given infinite time, you would be able to explain evolution entirely by natural selection. That's what he says in his later books. Um, then Weizmann, of course, also dissented from Darwin by removing the idea of inheritance of acquired characteristics by inventing his um, barrier. Well, the interesting thing about that is in the paper he published in 1883, just a year after Charles Darwin died. I'm not saying that's a coincidence or anything more than a coincidence. I got that the wrong way round. Um, but the interesting fact here is that when Weizmann introduced that idea and said natural selection on its own is sufficient, he actually argued it was not therefore necessary to demonstrate the fact experimentally that there was a barrier. It's incredible. Anyway, the fact is that later on, of course, when he was pressed by some rather wilder neo-Lamarckians on the possibility that, uh, well, particularly mutilation might actually be inheritable, he did his well-known and famous tail-cutting experiments showing that mice without tails, had them cut off, still bred mice with tails. Well, to respond to that bluntly, Darwin had already made it abundantly clear that whatever Lamarckism could do, it certainly did not allow mutilations to be inherited. After all, accidents in us humans don't lead to our children becoming and going around with exactly the same deformity. He, People understood very well that mutilations were not what Lamarck was talking about. So there's my answer, I think, to that aspect of what you've raised, Charlie.
Yeah. So, right. So I'm so glad you brought it to Lamarckism because the sort of acquisition of characteristics is sort of um, the crux of, what we're really looking at. How a phenotype can acquire new characteristics. In,
Yes,
you
indeed.
know,
Well,
in,
and we now know that the um, well, let me go for this one, if you may allow me to, because I'm keen on it. Um, Darwin actually collaborated for 10 years with a physiologist called George Romanes to try to prove his idea on how a Lamarckian process could work. Incidentally, he didn't use the word Lamarckian. Nobody did in the 19th century, but that's just another fact. And what he did was to collaborate with George Romanes, a student of my predecessor as professor of physiology here in Oxford, um, uh, John Burden Sanderson, way back in about 1880. He collaborated, that is, Sanderson's student, George Romanes, collaborated with Darwin in trying to see whether by hybridizing two plant species together, grafting them in effect, they could detect the transmission of tiny particles carrying the information necessary to go into the germline. They couldn't succeed, of course, because we now know that the particles that satisfy the criterion for being Darwin's particles, what he called gemmules, are the extracellular vesicles, which do precisely that. They travel from one part of the body to all other parts of the body, including down to the germline. And there are many articles now showing that. One that Daniel Phillips and I published just last year, called Bubbling Beyond the Barrier, cites more than 300 experiments now demonstrating control RNAs passing down through the germline. So I think we no longer have any serious difficulty with the idea that evolution gets speeded up by other processes. But very particularly, and this is why I'm keen on bringing George Romanes into the discussion, he wrote, incidentally, on the intelligence and mentality of animals, which is very relevant to your theme, agential action. And he did so because he thought, after his experiments, though they were unsuccessful in demonstrating what they were aiming to prove, it led, through interaction with Charles Darwin, to Romanes' very interesting idea of physiological selection, yet another process of selection to add to the ones that Charles Darwin himself had already generated. Now, I think that was what Charles Darwin thought also before he died. He died before anything could be published on that. It's all published in Romanes' books, published in 1886, Darwin and After Darwin. What does it do? It's beautiful. You see, Darwin realized that one of the difficulties about a Lamarckian process generating some variation is this. In artificial selection by humans, once you allowed the varieties to interbreed, you've no longer got the variation. It disappears. What was Darwin's big question, and the one he was trying to solve with George Romanes? What could prevent that happening? Now, the physiological selection idea is very simple. Even if varieties arise by random, I'm going to give it modern terminology now. the many genes involved in that variation and could be therefore responsible for the variation could sometimes have reproductive consequences, either psychologically or physically. And that's sufficient to enable the separation of species to happen. Remember what breeders have got to do. They have got to stop that happening to allow separation to occur. That must also happen in nature, and I think that Ramani's idea, developed, I think, with Charles Darwin on physiological selection, which is the word he used for it, might provide the answer to that question.
So, so that yeah, that brings me to another challenging thing I wanted to mention. So, assuming that Lamarckism is widespread, assuming that physiological selection is a ubiquitous or nearly ubiquitous, what do we do with results? Are okay, is there a problem with results that show that the acquired characteristics are have no effect on reproductive fitness? So, like, a lot of neo-Darwinists will point to, experiments that show that bacteria, under stress, sure, they acquire mutations, but those mutations are no more likely to cause them reproductive fitness than the ones that didn't mutate, that didn't acquire any new genetic sequence. And so they use those results to say, well, there you go. There can't be a level of physiology that's controlling the level of genetics.
Well, I think they've got the wrong idea of what genes can do. They don't control anything. I mean, it may seem like a remarkably silly statement to make, but I think it's true. Incidentally, just before I comment on that, two people, Wayne and Si, have got their hands up in case you want
Absolutely.
to. But I'll leave
Yeah,
you to make the.
we've got four
Oh, we've
hands
got
up. So
more up. Okay. But
yeah,
anyway, let me just deal briefly with that point. I think neo-Darwinism has got itself completely based on a truth that is not a truth, on an idea which is simply not a truth. Genes don't really control what we do. They, of course, enable me to have the proteins necessary to do what I do, and any other organism to do what it does. But does a gene determine in any way where I go, why I go? I mean, even at the fundamental level of how you use the capability that the gene for myosin, gene for actin, and so on give you to enable those muscles to develop, and for it to be the case that if you exercise, they will develop even more. Um, that in itself doesn't explain the behavior of organisms. So I think just at the very fundamental level, can genes control what we do? I think the answer is no. They only determine our capabilities, and we can't do physically beyond what we are capable of doing. That is obviously true, but I don't think it can really determine who decides to interact with who. That's the sexual selection idea, social selection, if you want to call it that, nor on a day-to-day basis what we actually do with all of those capabilities. I sometimes quote the end of page 18 of Futuma and Kirkpatrick's 600-page volume simply called Evolution. It's the only page in that vast book that is devoted to philosophy. And what does it say at the end of it? It doesn't even fill the page. This is their dismissal of philosophy. And what it says at the bottom of that page is there is no room for purpose in science. I think, my goodness, have they lived? Have they even ever asked themselves the question whether they have a purpose? Anyway, time, I think, for you, Charlie, to decide who comes up with their questions.
thank you so much. Yeah, I'm gonna I'm gonna call on the newest participants here just to be sure we get a diversity of voices. I know Wayne is dying to ask a question and Mary as well, but if it's okay, I'm gonna call on Cy and then John Kahneman, and then we'll have Wayne and then Mary, and so you guys go ahead in that order, and I'll be quiet. Thank you.
Well, first, Dennis, thank you for that wonderful presentation. It was really fascinating. In one of your early slides, you mentioned networks, and I'm wondering about the role of gene regulatory networks in the
Ah,
production of
yes,
stochasticity. Yeah.
there are none. You see, I come back to the point that genes do nothing. They are templates. And you see, the language of neo-Darwinism is so clever. They call them gene regulatory networks.
Right.
They are the processes by which genes are regulated, not the genes regulating the organism. But of course, it gives rise to that strange idea that the genes through those networks are regulating the organism. It just isn't true. Genes can't do that. They are templates. They enable a particular protein to be made. And, I'm sorry to keep on repeating it, but that is all they can do. So I never use the word gene regulatory network. I use the term functional network. Of course, RNAs are in there too. Absolutely right. They're also created by the genome, just as the proteins are too. But it is the integration of those processes at higher levels of organization where and let's face it, it's what the word we have to use, where the intelligence in the organism actually lies. It doesn't lie down at the level of the genes. The genes are just chemicals. And so I think that the central dogma of molecular biology needs to be treated as a simple chemical fact. DNAs need to be read to produce RNAs, and RNAs can be used by the ribosomes to make proteins. Full stop. That's all it can do. Any idea that it's somehow the case that there are gene regulatory networks, meaning that the genes are doing the regulating, is the wrong way of looking at it.
John?
Yes, thank you, Dennis. Wonderful presentation, and it's nice to see you face to face. There's a lot I could say, and I'll reserve that for the future when I know more about the group. But I follow the work of Robert Rosen and his MR systems in particular, and I see that as sort of fundamental to what you're saying. And genes are part of the encoding process in an MR system. It's encoding the choices and intelligence. But what I wanted to ask you specifically is on Lamarckism. I looked at that in the late 80s, and I was fascinated by the proposals of Baldwin. And it seemed to me
Yes,
that Baldwin provided the explanation for how
the Baldwin effect. Yes,
Lamarckian acquisition of characteristics could work. It's essentially a theory of learning.
yes. I think that's a reasonable way of looking at it, and the networks can learn, just as we now make artificial networks learn. And incidentally, I wonder whether I could deal here with also the point being made on the chat, which is that, well, I'll just cite it. It's from Green Fire. Wondering how the cytoplasm could employ liquid crystal metastructuring to manage and harness stochasticity. Phospholipid membranes are pneumatic LCs, and there are multiple opportunities for structuring of water that we now know are present in cells, from exclusion zone to membrane effects. I totally agree with that statement. The idea that all the water in that bacterium, 20 billion molecules, is free to move where it wants to go, is of course not what is the basis of my argument. It can't be that random. And I totally go along with the fact that much of it, well, we know, for example, that proteins can only fold by their water molecules coming in and occupying as bound molecules much of the structure. So a good fraction of that 20 billion is is used in that sort of way as part of the structuring of the cell. Totally agree with that. But of course, you know, we would have had an almost infinite amount of stochasticity in a bacterium if that was all there was to it. If all those molecules were free, they're not. But you don't need a lot of them to be free out of the many that there are for there to be still a lot of buffeting of the macromolecules, particularly causing DNA breakage, which is where a lot of mutation comes from, whether by radiation, by random movements of molecules within the cell, or in what other stochastic way. So it's a good opportunity to respond to that particular point on the chat.
Mary,
Can
do
I
you want
jump
to
in
follow
here?
that up? Yeah, go for it.
Great. Thank you, Dennis. First thing I want to say is I want to thank you for your tireless patience with us as really a cultural commentator and sort of historian of Western thought by dealing with the endless iterations of endless iteration of the central dogma, which I just recently reread at least part of the Voyage of the Beagle, and I realized that Darwin was a natural historian. He was a gentleman natural historian, really, in a lot of ways. He was traveling about, listening to folk tales and talking to gauchos, and in
Oh,
Argentina.
he was a
And
man way, way beyond how the way he is presented in neo-Darwinism. Yes,
so he had a lot of creative ideas. He was a creative
yes,
theoretician, but he didn't actually present a scientific basis for evolution of species that people think he did. And
no,
because he was coming from the a sort of a colonialist sort of empire building point of view, he wasn't allowed, his ideas haven't been allowed to grow. They've been stuck in a mentality. And throughout your career, you've really tried to help us to grow beyond that while honoring him. And I'm afraid that your message is still lost. So I wanted to make that cultural comment about what you're doing. See if you had any
well,
thoughts
thank
on
you.
that.
We have to do something about that, though,
You
because
really do.
the other side no longer has any arguments that I've seen that can effectively deal with what people like me are saying. And yet,
And exactly. And
who
Charlie,
still rules? Who
and
still rules the roost? They do.
it's so irritating. And we have Bill Miller in here and Henry Hang and um uh uh other folks who are working with trying to demonstrate that the stochastic story has got to be nonsense. There's got to be something like agency. It's simply the math doesn't work. But I wanted to ask you a question about this sort of like cultural
Yes,
transmission. It was my second question that I put in the chat. And
right.
I don't know if you can see it there. I have been observing through all of these talks and from reading what the agential biologists are coming up with that there's an awful lot of cells learning things and then teaching one another. And because we always have the transmission of the cytoplasm in division, I think that there's a huge amount of what I
Yes,
call cellular cultural transmission. And I'm wondering if you
absolutely so. Yes,
and
couldn't agree
Daniel
more.
talked to us about this. The last time we saw you, Daniel was talking about the vesicular transmission, but there's so many other
Yes.
pieces. But this also seems to me like it's stuck in kind of forgive the feminist. I don't usually think of myself as a feminist in a just dogmatic way, but this kind of idea that it's all about DNA, it seems like that's associated with sperm rather than looking at the cell and say the egg. For example, in sexual um, selection and reproduction and saying, well, what's all the stuff that the egg is passing on? What's the cell itself passing
Yes.
on? So
Yes.
your thoughts,
Well,
appreciate it.
it's phenomenally underrepresented in people's
Yes.
thinking, and it's crazy. in the debate with Richard Dawkins in 2022, and incidentally, on that, if you want a point by point response to the rather lame attempt that Richard has made in his latest book, The Genetic Book of the Dead, to recover from that debate, I've got the last chapter of the new book, The Pacemaker Channels of the Heart, devoted entirely to dealing with what he says in the Genetic Book of the Dead. That's just by the way. But now I want to
I'll have to read
come
that. You really are a
to
natural philosopher in the finest conception of that term, and I appreciate that.
well, thank you for saying that because I think, well, you'll appreciate a very important page or two in that chapter, which is it is risky to claim that philosophy has no relevance to science, because as soon as you open your mouth, you are using dead metaphors, all kinds of tropes that you possibly don't fully understand the etymology of, and without knowing it, putting yourself into a terrible situation. But now let me come to what Richard said in that debate, and it relates to what you're raising about the cytoplasm. He said, and I quote pretty accurately, I think, Dennis, we will carve your DNA into blocks of granite, and we'll keep those blocks of granite for 10,000 years, and in 10,000 years we'll recreate you. I said, no, you can't. Why not? Oh, Richard, where would you get my mother's egg cell as it was in February
Exactly.
1936?
Exactly right.
Now, you know, that egg cell had all the epigenetic stress factors that were the result of surviving during two world wars, in one of which my own father was nearly killed, in another of which I was nearly killed by a bomb that fell on our house. We lived under stress, our family all the way through. George himself, my father, was essentially a roaming street kid under total stress all the time. You ask the question, why do we find you get much better prediction of schizophrenia from the social class in which people are growing up? You get it precisely because of these communications, not all of which are purely social. They are within the egg cell itself as the cytoplasm. How could we possibly have ignored the cytoplasm? It's vast.
Because it comes from a woman.
If this was my nucleotide, could be whichever nucleotide you like, here
Yeah,
in a nucleus here in Oxford, the cell membrane would be way up in Aberdeen in Scotland.
exactly.
The cell
Exactly.
is
Yes.
absolutely vast. I did a
Yes.
calculation a number of years ago, asking the question, take the membranous structures, all of those folded reticulum membranes, the mitochondrial membranes, the ribosomal membranes, the nuclear membranes, there are membranes packed everywhere. How would you represent that in terms of digital information? It's very easy to
Exactly.
answer. At a resolution that needs molecular resolution, it
Yeah.
would have vastly more than 3 billion bits.
And to circle this back to the conversation about structured water. Structured water is creating the negative mold to many things in the cell, and in
Yeah,
all likelihood, as we can see from Luc Montagnier's crazy research with replication of DNA using structured water, in all likelihood, it's presenting an RNA-like capacity for countless facilities inside the cell. How could we possibly predict or record or observe all of that as well?
indeed. Yes. Well, you
So
said
anyway,
it.
so I think that I wish that we could have another 100 years from you to continue on with this battle. I think you've been very patient.
Well,
So
I'm afraid the
write
world
some
has
good
put up
books.
with me
Write
for
more good
nearly
books.
the world has put up with me for nearly 100 years already, because next year I'm 90. But that's another question.
Well,
No,
keep going. We need
I
more
am
good books
going to
from you.
leave as much of the argument
Thank
written
you.
down, carefully documented, and in addition to the book that's just come out, there are two more to come.
Can you tell us again the name of the book that you were citing to me? Because now I really need to read
The
it.
one that's coming out this month is The Pacemaker Channels of the Heart, and it contains around 4 of the 10 chapters dealing with what you would call the philosophy of biology, how I came to a very different position from the position I grew up with as a student at University College
Yes.
London way back in the 1950s. University College London was a centre of the neo-Darwinist modern synthesis. Fisher was involved with it. The goodness me, one of the Haldanes, J.S. Haldane, J.B.S. Haldane, sorry,
Oh, J.S. Holding. Wow. Yep.
was there until he went to India. He taught John Maynard Smith, and
Yep.
he was there until he moved to Sussex. Galton founded the chair of eugenics. Can you believe it?
Yeah. Well, Galton's part
That,
of the whole Darwin clan, and they were all, you know, they were all very much focused on this sort of top-down control centers sort of focus in biology.
yes,
And and eugenics as a global good.
yes.
If it works for
Oh,
cattle, why wouldn't it
they,
work for
they fully
humans?
believed it. And
Oh,
and
sure.
Julian
Yes.
Huxley repeated it in 1963 in the second edition of the Modern Synthesis. He
Oh,
said
yeah.
there's a role for eugenics to improve the human race.
I have a book called Home Eugenics, and it's about it's about making good choices in the way that you raise your kids that will allow the strongest to succeed. It's just really, yeah. So
Awesome.
anyway,
Well,
I want to
it's
call on Henry since you brought
Hey,
up
wait, hold
Galton.
on, Barry. Wayne's had his
I
hand up
could.
this whole time. Let's let Wayne go, please.
Okay. Oh, sorry. That's right. Sorry, Wayne.
We'll come back to Henry. I want to hear from Henry as well.
Yeah, yeah, sorry. Yeah. I mean, I could say a million different things. Dennis is kind of my brother from another mother in different generations. If anything, I'm actually more radical than Dennis in some areas. I've also been fighting against this for decades since I was a grad student, before I even switched into mathematics. It never made any sense to me. None of it. First of all, well, yeah, I did want to touch on, on Dennis's last slide, when he's talking about constraints over many different levels. Our last speaker, Terry Deacon, would have agreed 100% with this. This is most of what his work has been about for the last 15 years or so. When he talks about emergent constraints and he talks about teleodynamics, and especially when he talks about the paper he wrote with Kucher-Finis, teleodynamics and dynamical depth, where he looks at these types of emergent meta constraints at different levels, actually over many levels, where they do take advantage or use the underlying dynamics almost kind of in an opposite way. So we were talking about, and I kind of want to tie in a couple of things here. The idea about random mutations. Really, to me, random mutations are probably not even important. They're not an important mechanism in evolution at all. But they are important.
well, if I can just interject there, Wayne, for a moment. I think natural selection, as originally conceived by Darwin, as the opposite of artificial selection, is rarely actually relevant because, as I said earlier on, the great majority of our environment now is other organisms. They're capable of choice. There's not much natural selection left.
I don't think there really is any.
Sorry, I'm interrupting you.
Oh, that's okay. I mean, I got sidetracked by all the other commentary. I didn't even get to kind of say what I was originally thinking. Yeah, I mean, one problem I see is if you get bogged down in a particular mechanism. So yeah, whether it be the digital memory of DNA, which is just one of many engrams that can be used by the cell at higher levels to achieve various goals. It's just a tool.
Yes.
The same is probably true of structured water. And they're all examples in many cases of just acceptation. Once you have a system that's an agency that has intrinsic normativity, is capable of having goals, it's going to use whatever it can in any way it can to achieve those goals. And often in who is the other pragmatist, William James, where his definition of intelligence was arriving at the same endpoint by different means. This is what organisms do.
Well, thank you for those comments, Wayne.
Henry,
Of the hands up, I think that just leaves Henry. Is that right?
That's right.
Yes. Oh, hi, Dennis.
Hi, great to
very-
see you again.
Very
Well,
like to see you again. Yeah.
Let me just say, this is the author of Genome Chaos. Am I allowed
Yes.
to publicize your work, Henry?
Thank you. Thank you, Dennis. So
Yes.
like every time listen to you, it's just always get very excited. So anyway, so I have two comments also would like to ask you advice on it. So the first thing is because the organism have the own agency, the selection, so it's constantly interaction type of selection. So we use the terminology mutual selection. How do you like this one? So the mutual selection actually is a gradually so-called evolvability is gradually increase. So initially mutual selection they have lots of physical feature dominate, but then gradually with the information contact increase. So that's actually the mutual selection become dominate. For example, maybe we are going to someday go to leave the Earth, for example. That's ultimately mutual selection behavior and decision. So that is one thing I will discuss this more and listen to you advice, some other day. So another question is, we actually, after we separate macro and micro evolution, we realize that Darwinian, most of them is a microevolution. Therefore, the microevolution fundamentally is conservation, is a constraint. So in that sense, the physiological and the biological is a powerful mechanism for constraint, which initially when the system formation, the actually winning from random. But after the formation, the constraint is keep away from random. So, right. So in that sense, the physiological also become more and more important in terms of ensure the trend of evolution. But maybe in the future, after artificial, intelligence, after the technology selection more dominate, they could actually overtake some of physiological selection power by the last stage of selection, which is culture technology selection.
I think you have to be right, Henry. Yes, on that. In the end, it's now cultural, isn't it? What else is driving the madness of this planet at the moment?
So if that's the case, we need to acknowledge the evolution on the Earth actually is a process of increase the complexity.
Yes.
So another thing related to this is because increased complexity, therefore every species is replaceable as long as the information is passed to the next carrier, right? So therefore, very interesting enough is the intelligence could be in the middle of this whole process, become more and more dominant as such. So right. do you think this is a fit? What you have talking about how to use the stochasticity in such a way is actually reduce all the probability, and therefore they can ensure the increased degree of complexity. So therefore, we think in the evolution, we need a 3 level. First level is information package themselves. Second level is carrier themselves, including the information. Third is ecological level. So all the 3 level is constantly moving. So now the species, you cannot move back because the ecosystem already no longer there. So when you move back, your biological meaning also changed. So that's the way we actually thinking this, how the different stage and the information get more and more complex and the carrier get complex. Rather than the previous people thinking evolution is no direction, we think it is have direction. And at the in the future, the intelligent part, cognition part, consciousness become dominant, will completely change the natural selection in the past 3.5 billion years. Is that fit the way of you are thinking? Yeah.
Well, once you get, Henry, to, as you're implying here, once you get to conscious selection, you're precisely on what I think Darwin was getting at when he talked about sexual selection or social selection, as I would prefer to call it, because I think it's both. But he actually specifically said of the birds that they consciously select. And he used the very word conscious. Of course, the neo-Darwinists have written this out of Darwin. When I had that debate with Richard Dawkins in 2022, he said, you know, Denis, you are a late Darwinian. I said, no, Richard, it's there in the very first book, The Origin of Species. The Lamarckian idea is still there. I said to him, I'm sorry to say it, Richard, but Lamarck is back, and Darwin was a good Lamarckian. And he was utterly shocked. He's never recovered from that because his argument in the Genetic Book of the Dead is very poor. He doesn't even understand the difference between active and passive causation. I mean, you know, this was introduced by one of the greatest scientists ever, Aristotle. Aristotle wasn't just a philosopher. He was a very good early biologist. He introduced many of the ideas that were taken up later by much later biologists to form the basis of what we know today. Even had at least preliminary ideas of the evolution process. But his main and fundamental distinction was between passive causation by form and the mere structure of something existing, whether it's a cell or a great big galaxy. And it corresponds exactly, which is why I use the word biological relativity, it corresponds exactly to what Einstein was talking about in his general theory of relativity. That is, a huge structure distorting, in his case, the background, space-time, to cause light to bend as it moves through the universe. You know, it is, I think, very obvious that this distinction between passive and active causation is utterly fundamental. Nowhere in any of Richard's books or in any of the books written by neo-Darwinists can I find even a recognition of that fundamental distinction. And yet it's a mathematical necessity. Just ask the question, how could you ever solve the equations for any particles moving around in a cell without the boundary conditions? It's a mathematical impossibility. Without those boundary conditions, there are no solutions. Now, that's just the structure, that's the form. The sequence in DNA is also a form, it's a structure, it's a passive cause. I don't know how often I'm going to have to repeat this, but I'm happy to repeat it endlessly until the other side get the point. And I'm hoping that the latest book, The Pacemaker Channels of the Heart, although it doesn't say it's immediately relevant to these arguments, it is, and particularly the later chapters in the book.
Thanks.
But thank you for your points, Henry. And I've always enjoyed your work on what you call genome chaos and all the various processes that you've carefully documented on how the genome is stirred up, to put it up, I think in the terminology I would use.
Nice.
And
Thanks.
I
Yeah.
think that just leaves William, doesn't it?
Bill.
Bill Miller.
Yeah,
Yes.
Bill Miller. Bill Miller,
Yes.
it's great to see you finally, since we have
Great for me to see you.
emails.
We've never managed to meet, have we?
No, we've traded emails for about 8 years. First,
That's right. Yes.
I think you're one of the very great individuals in biology. I think your work is an essential stepping stone in a very necessary shift away from neo-Darwinism. You gave me a great boost in an initial article I'd published in Biology in 2016, Cognition,
Yeah.
Information Fields, and Hologenomic Entanglement. And your kind words have really spurred me to think deeply and coolly about evolutionary theory. And as you know, I cite you all the time. So you know how much I admire your work, your concepts of a no privilege level of causation, harnessing of stochasticity. But you said very early on that intelligence is a major living factor. And my feeling is that there's a simpler way for us to approach biology, including genes. Once it is granted that life is intelligent, then selection becomes cognitive selection. It is intelligence selection, be it sexual selection, physiological selection. The key, from my perspective, is once cellular cognition is embodied within the cellular form, for whatever reason it wasn't, which we do not know the why or how,
No,
that everything that spills forward is intelligent cognitive selection. And then we understand the place of genes, as you've carefully explained, and I agree with completely. Genes are tools of cells. They are forms of retrievable and deployable memory for cellular decision making and problem solving. I'll say, lastly, that you and I are in complete agreement on central dogma. If you remember, you were the managing editor for a paper on revision, 21st century revision of the central dogma that František Beluška and I and a couple of others.
Yes, indeed, I remember. Yes.
Anyway, I would finish by saying that I think it's important, once we grant that life is intelligence, and I'll make the further argument that natural selection is cognitive selection. The natural selection that Darwin was talking about is filtering, environmental filtering, like Jim Shapiro would talk about. Then there's another form of contrariness to the central dogma, which is unilateral flow of molecular flow, the DNA to RNA to protein.
It's always two ways. Yes. And I can't understand, how is it that the world of physics moved from one way to two way once it accepted Einstein's relativity principles, both of them, the special relativity idea, and then much more significantly his general relativity, and then the transition that arose because of the discovery of quantum mechanics. How is it that biology went in precisely the opposite direction? It's unbelievable. I mean, the very physics of the universe is now understood to be at least two-way. You can't interpret the general principle of relativity or Einstein's, well, we get all muddled up with the terminology, don't we? Theory of general relativity, which is what enables his concept of space-time and its distortion by huge objects in the universe to come in. You can't even talk about that if you stick to a one-way process. It's just not possible.
Yes, in the end, it comes down to a simple proposition, which mirrors exactly what you just said, in a different form. Once it's understood that cells are the basic building blocks of all living organisms, then you have two choices. Cells are either automatons, which would be Dawkins's point of view, robots,
Exactly. Yes.
or they are intelligent, measuring, decision-making agents. And those are the two big decisions, because once they're agents, you understand the origin of variation. And now you understand evolution in a better way.
Yeah. Simple, isn't it?
Thank you, Dan.
Are we going to be able to convince them that? I have a book already called Fight for the Future of Biology. That's my preliminary title of it. And it's ending. It's with a publisher at the moment. I don't know whether they will take it. But its ending is precisely to say, what on earth are we all arguing about? Because I list the various things that we've discovered that I think are incontrovertible, including the fact that DNA itself can't be the blueprint for life. And I then say, well, what is there left? Well, there's only two things left, I think, out of all of that. One of them is the idea that there is purposive selection, which is Darwin's sexual selection, which I think could also incorporate what I now call physiological selection. So I think it's perfectly conceivable that there could be a resolution of all of this. I call the last chapter of that book, incidentally, Resolution. And I don't really think there's much else to argue about at the level of the detailed biological facts. And I don't see why the other side can't see that. It's time to throw the towel in. It's time to revise the textbook. Well, that's what we're going to do. There is a meeting on evolution coming up in January, which we control, or which we designed, let's put it that way. And that's going to be held in Oxford on January, very early January of 2026. And that's going to be followed up. Of course, there will be video recordings of each of the main presentations at that meeting, but it's going to be followed up also with a very determined project to produce the teaching material for the young. Now, I'll tell you what I've been doing mostly during this year. I've been all over the place, to South America. I've been in East Asia. I've been, of course, within various parts of Europe. I've been interacting time and time again with young people. I lecture now at one of the top institutes, university institutes in South Korea. The audience I get are their faculty, but also their great young students studying. And I get fantastic reactions. You see, I think, yes, there is a group of people who still control the main funding agencies, still control many of the journals, but not all by any means. Notice that paper with the diagram of extending the so-called central dogma was published in the journal Evolutionary Biology. They're no longer in control of everything. I think we should feel a bit encouraged. There's a bit of a tipping point here. And I'm hoping that if I can get this other new book, not the one that's coming out this month, if I can get that through into a good publisher, I think the challenge will be there. Can't we agree on this list of biological established facts? And if you do, what is the argument about? Why do we continue to have to misrepresent Lamarck and misrepresent Darwin too? I think it's one of the most scandalous things that's happened in the last 80 years, all the way from the modern synthesis in 1942, that we have misrepresented two of the great biologists of the 19th century, Lamarck and Darwin. Sorry for this. Okay.
Yeah.
So what do we do now, Charlie?
Well, well, I have one more question. We are getting close to time, but we've got a few more minutes. I so um I was reflecting when Bill made his comment about the measuring sentient cell being the fundamental unit of agency. I was thinking about your theory, Dennis, and wondering how you would deal with this question. So I'm going to try to make it as clear as possible, but it's I hope I can get it across. So when we talk about individual cells or organisms having agency, I think what we mean is their behavior is shaped by the world around them. They're sensitive, open systems.
Yeah.
And they have a domain. They have some sort of cognitive domain that can be shaped by events. But to me that conflicts with the idea of survival and reproduction as the teleology toward which life orients itself. Because to me, survival and reproduction is not in the domain of the individual. It's not something an individual can learn from. They can't orient towards it. They can't be affected by it. That's like a lifetime reproductive rate, or whether or not you survive is not an event within your lifetime, right? It's an event in the domain of the lineage. Um, and so I wondered how you dealt with that in your theory, how you think about the cognitive domain of an individual versus a lineage. Is it?
Well, I have a very simple view on this, Charlie. I mean, and it was expressed earlier on by Henry Hane. We were already into a period in which cultural evolution is almost more important than biological evolution. And I think that's correct, isn't it? As I said earlier on, the environment is now that of mostly other agents in the environment. There's very little left that is actually purely physical in the sense that Darwin was imagining when he introduced the idea of natural selection. It was an idea which was that the environment was a purely passive filter. That's exactly what natural selection is, a purely passive filter of the survival of the fittest. But in a context in which we are generating cultural tools and processes which can harness the way our biology is moving, I wonder whether we've not reached a point where that is almost more important. And I think that's going to be even more heavily true as we develop to use artificial intelligence in the various forms in which it's being developed. I supervise a graduate student at the moment working on artificial intelligence, and the aim of the work being done there is how do you make it compatible with human values? How do you ensure that it becomes useful to us as the development of our culture? And that's going to speed things up enormously because, as we know, the great advantage of AI is how quickly it can do what it does. You just go online and ask what I'm going to be doing next, and it will come up with all kinds of ideas of what Denis Noble is going to do. The problem is none of those may be right. you see, I don't know what I'm going to do next. So how can it know? After all it does is to find out what's on the internet. But look, I'm joking a little bit now. The key, I think, is that we've entered a phase of evolution in which cultural developments are almost certainly going to matter much more than our own physical evolution. At least that's the way I see it, Charlie. In which case, what we call intelligence and what we call the development of our cultural values is going to matter much more than almost anything else.
Gotcha. Looks like Mary has a question. I think you're muted. There you go.
It's the same question. Dennis, I really don't think that what you just said is different than what I asked earlier about cells literally gathering packets of information, memes. And I used the Daniel's example of the lipid packets that encode
Yes.
RNA. That pass on RNA, which comes from the experience of the parent cell. I think that it literally is cultural evolution, but it's a holographic or fractal um, iteration. I agree with Michael Levin in that I don't think there's any such thing as emergence. I just think there's cellular capacities writ large. I don't see any difference between cultural transmission in individual cells and transmission between cities, humans, tissues in the body, um, biofilms. It literally seems like it's a capacity of the cell. And I'm working with a
Yes.
group of biologists right now to create what we're calling holonic biology, looking at all of the things that cells do, starting with what I call a life homologies approach, how cells talk to each other, build cities, lie, eat, move about, build relationships, how working with Fred Provenza, how sheep do this. He spent his career studying grasslands ecology and whole animal biology, and how the same things that cells are doing, we see in all of these different levels. And the principle that we don't need to be less anthropomorphic, we need to be more. And if we understood ourselves as primarily culture creators and transmitters, a lot of our questions that we're so hung up on in evolution just go away. They're not interesting anymore. That's my thought. It wasn't a question. Sorry for the spot.
Awesome. Henry's got his hand up. You'll have to unmute Henry.
Hi, Dennis. I have a last question for you. Yeah.
I'll try to kill the phone call. Just a moment. Oh dear. Well, it's killed itself. Sometimes it's not necessary. People get fed up.
Okay. Okay.
Sorry about that. Over to you, Henry.
So for a long time, we actually kind of confused about the function of the microevolution, right? So the idea is for a long time, population genetics thought, the gradual accumulate, so the gene, beneficial gene, the spreading in the population. But the recent year, when people did the large scale trace up, the microevolution never accumulate. So if they are never accumulate, we actually predict they're not going to accumulate. We think the gene mutation come and go, as long as you have the framework, so they don't care lower level of stochasticity, right? So that is going, as long as you have the framework, you don't care who is, involved this whole process. That's why majority of the gene, actually the function can be compromised without, have the function consequence. So therefore, the question is, if that's so, why the little microevolution at the first place?
Well, we don't.
So
don't
maybe
really.
we can say simply just for the survival, right? Okay, for survival it's good, but when the population becomes so dominated, the population will replace. So if you put all the story together, actually the nature doesn't care individual species either. They only care the information continually pass, continually flow. So I think maybe that's the soul of the evolution. So how do you like this type of analysis?
Yeah. Well, that's a good statement, Henry. Don't disagree with it.
We'll continue thinking about this one. Yeah.
Yes, I think so. Yes. But I don't have any late night. Well, here in England, it is late night. Well, not so late, but it's nearly 6 o'clock. Let's put it that way.
Yes. Okay. Yeah.
Can I make a quick comment for Henry here? So one of the fundamental assumptions basically of population genetics, I mean, this is kind of their ontology. They're basically assuming that there is some kind of a fixed fitness landscape, or basically the environmental to a first approximation is constant, but it isn't. The genes and every other thing that gets passed down is an engram that constantly has to be reinterpreted and reenacted in current context. So why would they accumulate? They just get reinterpreted, and.
So when you are right, because we have to remember the mechanism of natural selection is based on this accumulation motion.
Yes.
So if you think there's obviously there's no accumulation, then we had a problem, right? So that's because in biology, I found it's funny. Is everything you in the certain domain always makes sense, but if you put all the domain together, then the paradox appear. So that's a thing we like,
Yes.
That is right. There's no privileged, there's no privileged level of I guess of operation. I mean, any particular level you look at, it's going to be important depending on the question you ask that you look at the right level. But there is no privileged level, whether it be genes or whether it be cells or whether it be organ systems or anything you'd like.
But at another hand, you gradually, if you're thinking the information management, so it appears different level of scale that have different dominant role to regulate the stuff. So, so for example, if you're talking about intelligence, of course, the brain, everything become important. But if you're just talking about energy production, right, it's totally different to the prioritize. So we actually try to prioritize from the history of evolution. So every stage, you actually can find one of the key evolutionary mechanism to push the information flow forward. But after that, it's no big deal, become biological routine. They just use biological process to finish this miracle previously. So therefore, the evolution is many stages, each stage with a major prioritized principle. So if you think in that way, we did not actually prioritize in terms of the life scale, but we prioritize in terms of evolutionary history, where the information from first cell and to the multicellular and the superstructure of the society. So every time actually they do have prioritized the mechanism. Is that make sense to you guys?
Yeah, that makes sense.
Yeah, Henry, so if you look closely enough, everything really is molecules banging against each other, but you're ignoring really final cause. That's the problem when you ignore philosophy. We've broken everything down, and most modern science are locked into this mode where they're looking for the proximal cause and ignoring the entire big picture, right? What problem space are you working in, whether you're an organism or you're a person studying it, kind of tells you what levels of organization you should be looking at.
No,
appropriate.
We actually is not doing that. We're thinking if the information flow is a major, the force, then how this can be achieved during different stage of evolutionary. That's actually the picture is emerging is because, every time they solve the key problem, the first they solve how to form the system, therefore the first cell membrane, and the second time they don't have enough energy, therefore, mitochondria are captured. And then they have the cell differentiation, so therefore epigenetic become dominant, even though epigenetic can found from bacteria, but no big deal at that stage, right? So gradually the animal and the brain and society and the new technology, so each of the stage, they actually have some of a very powerful principle overshadow others, right? Because energy is very important, yes, but then energy is no longer important. We have so much energy in the, the cellular energy is no longer the key stage from, the bacterial to eukaryotic transition, for example.
Henry, I'm sorry to jump in. I apologize, Mary, but exactly pertinent to what you're saying. You're talking about information flows. How do you reconcile macro evolution and microevolution? And there is one way to do it that is not honored in biology, but is a very important principle. Cells have states of preference. Every single cell has a specific state of homeoretic equipoise, dynamic state, like a Pascal and Proce's dynamic kinetic stability. Every cell has that, and it is a preferential state. It is a state of satisfaction. And when you begin a system with intelligence, a distinction between the outside and the inside, maintaining that internal equipoise is preference. And once you have preference, you're no longer, Charlie, with reproduction and survival as the dominant force in evolutionary flows. You're with achieving states of preference either individually or the collective form. And actually, we've conducted a huge experiment without planning to on exactly how powerful is states of preference versus survival and reproduction. And we've done it through birth control pills and the dropping birth rate in all developed nations. We know that people would rather have sex for sex than for reproduction. Reproduction is an epiphenomena of states of preference. And we've reversed this because we've never recognized that the state of preference begins with the unicellular form because it is individually intelligent. And biology shifts dramatically when you understand that.
So let me just respond back. It's quite interesting. We are actually not against what you are talking about, but we actually visualize the macroevolution is create such a boundary of what you are talking about, the best status. And then the microevolution is grow the population that actually fulfill the different landscape. So therefore, the macroevolution always create system, microevolution always grow up the system in such a model. So therefore, the including what you are talking about, for example, but when you mention, the whether or not have kids, that's another beautiful example of the culture force is over the biological principle as such. So therefore, you can say the different stage actually is different principle play more important role as such. So we actually agree with each other in the way. Yeah. Thanks.
So one important thing to say here, I have my hand up, sorry, is that, one thing that you have to keep in mind here is that all these levels are highly constrained by the upper levels, and there's give and take between both levels. So yes, a cell does have preferential states it wants to maintain. However, when a cell is part of a tissue or part of an organ system or part of an organism, it's highly constrained, and it's also, in a certain sense, a lot of those needs are being met by the higher level. So it doesn't actually actively have to worry about maintaining those states. All right, so.
In fact, so for the evolutionary innovation, most of the time, is all of a sudden you create a new higher level. So that's what we were talking about. So so every time the higher level is achieved by new organic coding. So every time the complexity is achieved by the more coding and more coding, but the coding right now, your cultural coding, the code overpower the biological coding. For example, people said, okay, I want to have, the more woman or whatever, like the very thousands years ago, but now we are going to put you into jail because that is so powerful, right? So as a bio machine, the behavior of of human sexual behavior is no longer just for the sake of the reproduction, right? So those you cannot imagine in the early stage of life form. So therefore, the law is different every time along the way to go in. So use this idea to review the evolutionary theory. All of a sudden, we can put every scholar's theory along the line of the evolutionary history. It's fascinating if you see that way. So therefore, Darwinian initial idea, because the majorly focus on artificial selection, that is very late stage of evolutionary phenomena, which therefore cannot explain, before human appear, because this totally different world we have to explain. So if you use that way to thinking, I think it also help for understanding the cognition, how powerful they are. Now we have very powerful, but it's not as powerful as in the bacteria as such. So that's the whole point of, to see the slightly different.
I have to jump in here.
Go for it.
There really is an emerging conversation about information as sort of and ontology, and bringing in that final cause. And I'm hearing Aristotle referenced over and over again in different folks, from the folks who study Rosen to Mike Levin is now talking about information and final cause. And I think that this group is now coming to information, final cause, Aristotle, and culture. And I just want to notice that this is a cream that's rising to the top that I feel needs to be explored more fully. And dare I say it, for us to begin thinking of ourselves as natural philosophers once again. This is what happens as we're going through a scientific revolution. We're no longer acting as technicians, as bench scientists, underneath the weight of the central dogma. We're trying to emerge something new. And I just wanted to make that cultural commentary.
Which Dennis set a good example for us to follow. Yeah.
Awesome. Well, guys, we've pretty much come to the end of our time, and just to respect everyone's schedules, I think we'll have to bring it to a close. But thank you so much, Dennis, for being with us in this time. And
That was great.
Yeah,
we've
really
all-
good, Dennis. Thanks a lot. We hope you'll join us again.
Great privilege to be invited to address this clearly very interesting group. Yes.
Well, we strive for that.
Just
Okay.
ordered the book.
Just ordered the book.
Absolutely.
Yes.
Absolutely. And
Not exactly cheap, I have to admit.
yeah.
But it's produced by what they call the monograph series. Anyway, I will say goodbye to everybody. Time for me to get some supper and get to sleep. Bye bye.
Thank you so much. Bye bye, Dennis.
Bye bye.
Yeah,
Bye now, Dennis. Bye,
and
Charlie.
everyone who joined for the first time, please come and see us again. Thank you so much. If you don't have my email address, I'll put it in the chat right now. And if any of you want to reach out to me to join later, I'll be happy to oblige.
Great job, Charlie. Thank you.
Right. Yeah, Mary. Thanks to you too. So you're going to be near Vermont soon, huh?
I am. I'm in Maine.
Okay.
2 hours away from you.
Awesome. And you're coming through Brattleboro, you think maybe Saturday or Sunday?
Yeah, we Philosophy Farm's really coming together. One of the presidents of Marlborough has donated us his philosophical library. It's like 10,000 volumes. So now we have to renovate one of the barns so that we have a place to put the library. So we're just in the throes of renovating the property here. It's insane. I'll live.
Totally. Great.
So
All
it'll
right.
be good to see you.
Well, yeah,
So
we'll reach
I'll be in touch
out.
with
Yeah.
you soon.
Let's have some coffee, when you come through.
It'll be great. And I want to meet Josh too.
Okay. Awesome.
All right. See you guys
All right.
later.
Okay. Bye-bye.